Alright, so this is the DNA. This is helicase, which is the enzyme that's going to split the double helix DNA into the leading strand and the lagging strand. So the DNA enters through, the helicase is ripping through the DNA right now or whatever, and right here through the replication fork is where the two strands of DNA become the leading and lagging strand. And then eventually they go through this other enzyme after the helicase called DNA polymerase, where the leading and lagging strand of DNA bond with a corresponding complementary base. Alright, so as the DNA strands are broken up into the two DNA polymerase, the leading strand is quickly bonded with its complementary bases and then continuously pulled through the DNA polymerase. As the leading strand is being copied, the lagging strand is also being copied in Okazaki fragments, which means that only a few copies of them copies can be made at one time, and it can't go through until the polymerase is released from it, until it releases it, and then it continues on until both strands are fully copied and have complementary bases. So as the leading strand is finished being copied, it enters into the RNA polymerase, where it then turns the leading strand and the complementary bases into RNA. So now that all we have left is the leading strand, because the lagging strand moved on to go do other jobs in the cell, the leading strand is going to move into the RNA polymerase, and by 3 it's going to line up with the top part of the leading strand and then create the mRNA strand in 3 pieces. It lines up, and then those pieces just continue to move along as the strand moves throughout the RNA polymerase. Okay, so after leaving the RNA polymerase site, the mRNA that was created previously is the only thing that starts in the ribosome. So a tRNA methionine is going to start in the P site, and it's going to wait for the ribosome to go along, and then it's going to attach to the start codon, which is AUG. After the original tRNA has been added, the next with the corresponding codon is going to join, where then the first amino acid joins to the second to start creating the polypeptide chain, and this moves over where the tRNA now exits through the E site, and the next tRNA joins at the A site in order to start the whole process over and help to start forming the polypeptide chain until the whole protein is created. Once the last tRNA has been copied in order to create the chain, a stop codon comes along to stop the polypeptide from growing, where then the polypeptide goes and folds around in the cell to go complete another task.